Okay, good afternoon, everyone, and I'm delighted to welcome you to the latest BMK webinar, How Accuracy Long-Read Sequencing and its Implications for Genomics, Transcriptomes, and Metagenomics. I'd like to thank you all for taking the time to attend today's webinar. And today's webinar will last for around 30 minutes with 5 minutes for question and answers at the end. You could submit your questions via the Q&A button at the bottom of your screen or raise your hand. We are looking forward to receive your questions. And we will share the slides in a recording of the webinar by email. Now I'd like to introduce the speaker of today's webinar, Wilson, who's our Business Development Manager of Global Service Department. In this webinar, he will share the basics and the principles of long-read sequencing technology and the implications of HiFi reads, such as the de novo assembly, the variation detection, and full-length transcripts and amplicon sequencing. And this webinar will illustrate how HiFi reads are empowering various areas of life science research. Hi, Wilson. Over to you.
Hi, good morning. Good afternoon, Emma. Thank you very much for the introduction. So today I'm very happy to give a talk on this topic on the long read sequencing, which is I think is very interesting for many scientists in Thailand, because most of scientists will more familiar with the short read sequencing. But for now, I think long reads has become more and more important and can solve many limitation for the genomic research. So today I'm going to give a talk on the high accuracy long read sequencing and its application for genomic and transcriptomic and metagenomic research. I'm going to give a talk in English, but for the Q&A, I'm happy to take questions in Thai. Okay, so let's move on to, so basically, let me introduce BMK gene or biomarker technology. Basically, we are a sequencing service providers which own several sequencing platforms from Illumina's 10x Genomics, PacBio, Oxford Nanopore, and also mass spectrometries. And importantly, we also have an in-house bioinformatics software which can serve in many applications from genomic, single cell, genomic, transcriptome, and so on. So we are a one-stop service sequencing provider then. And we have been in the market for many 10 years, and we have more than 500 staff, have more than 60 national patent of invention, and also the more than 200 software copyright. And we are very proud that we contribute in many publications in the highly prestigious journals like Nature, Cells, Plant Cell, and Science as well. So let me start my talk about the evolution of sequencing technology. So in the past, we are familiar with the first generation sequencing like Sanger sequencing, which are very good at providing high accuracy. But the fragment is quite short. It can sequence up to 1,000 base pair. However, Sanger sequencing still remains relevant in many research applications. After that, the next generation sequencing has been developed by Illumina, and they can sequence like many short fragments simultaneously in parallel manners. So this can accelerate the research in genomics a lot. And recently, the third generation sequencing has been developed by PacBio and Oxford Nanopore. These two platforms are very useful, which provide a long read, which can solve many limitations associated with the short read sequencing, which I'm going to talk to you in this presentation. So in this webinar, I'm going to talk about the basics principle of the long-read sequencing by PacBio HiFi and the application of this sequencing in genomic, transcriptomic, and metagenomic studies. Uh, let's compare between short read and long read HiFi technology. For the short read, they provide relatively short fragments, but it can provide like kind of high accuracy, like 99% accuracy, which is very useful for many applications already. However, recently the long read sequencing HiFi has been developed, which can do long read like up to like 10 12 kb, and with a high accuracy as well, comparable to the short read technology. And other long read technology can provide much longer read length, but the accuracy will be kind of lower than the HiFi as well. So uh, basically, HiFi read can provide very long read and high accuracy at the same time. So if you can have a look on the scheme in the right hand side. So when we read, we do a sequencing of the reference gene kind of samples using short read. We need to incorporate the short read map with the reference genome. However, in many cases, there are sequencing gaps, which we don't get the data from the short read. So the HiFi read has been developed to solve this kind of problem. So the long read can map uniquely and can span the very long variant associated with the research question. So you can see that the long read by PacBio we can call like smart sequencing. It is a single molecule real-time sequencing. So you can see that the DNA is prepared and then sequenced in the small well in the flow cell of the PacBio instrument. So you can see that the dye incorporate with a different kind of nucleotide like A, T, C, G will provide a different color. So the instrument can detect the emergence of the different colors. So it can be translated into the signal of each sequence of nucleotide. So this is the principle of smart sequencing. And PacBio has several platforms like RS2, Sequel, and Sequel2, which have different throughput and also yield. So then how the HiFi read is generated? We started from the high quality double-stranded DNA, which we ligate the adapter sequence at the both end, and then we add an unilink primer and DNA polymerase, and we can put this into the flow cell. In the flow cells, the DNA polymerase is working, and then it synthesizes a new strand with it used the fluorescently labeled nucleotide, which generate different signal. So the instrument can read the nucleotide sequence from that reaction. So this provide the circular the in the circularized DNA, it provides sub-read, which the bioinformatic can extract and it provides a consensus sequence, which has kind of very high accuracy, up to 99.9%. So the advantages of smart sequencing, like it provides kind of very long read compared to the old technology. Most of read can higher like 10,000 to 25,000 base pair. And it also provides the high accuracy as well. So majority of the reads have the high Q30 score. And it can provide the uniform coverage as well, because this technology can work very well both high or low GC content. So have a look at the PacBio HiFi reads. It provide kind of long read from starting from 10 kb or 15 20 kb, and with the high accuracy as well compared to other technique like ONT long read or ONT alpha long read, which have kind of inferior accuracy in this case. Using HiFi read technologies can provide a very broad application like genome assembly, which so far PacBio HiFi remain become the majority in many genome assembly projects. Metagenome analysis as well, and comprehensive variant detection, they can detect many types of mutation, and also like the structural mutation, which kind of challenging for the short-read technology, can be done using PacBio. And more recently, like the transcript sequencing, we can the PacBio technology can make the full-length transcript, and also can differentiate different isoform very nicely, and targeted sequencing as well. So let's have a look on the first application for the PacBio CCS, like de novo genome assembly. We know that we working with many species of organism. So many of them has a complex and giant genome. They could be autopolyploidy and allopolyploidy, including repeat and heterozygosity. And all these features complicated the short-read technology. And so that's why the long-read sequencing technology has emerged. So this technology has been used in many genome project, like recently, like telomere to telomere human genome project that I'm going to talk in the next slide. So the strategy to do the de novo assembly, like we usually start with the survey of the genome by short-read sequencing, and then we do the long-read sequencing to do genome assembly. Polish and correction is an can help to polish using short reads as well. And then we do the chromosome assembly using HiC. So like in this table, you can see that for the de novo genome project, it usually involves many types of technology from short read, long read, and also HiC to get a better, the more complete picture of the novel genomes. So for the PacBio, we can do the sequencing in many challenging area, like repetitive sequence, which are kind of difficult for short read to assembly this region, but long read it can span through this region. And also heterozygosity, like diploid organism which have maternal and paternal copy of DNA. The long read sequencing can easily distinguish between these two types of DNAs and provide very clear genome graphs. And this is an examples that HiFi read can provide very high accuracy for the variant calling. Like for example, STRC genes in human. If we use just Illumina sequencing, we will end up with some unmapped region. This might be because this region is less accessible to the short read. But in HiFi read, we can give a very nicely coverage across the region of this gene, and we can clearly call the variant. However, for ONT technology, they can span over the whole gene as well, but due to low accuracies, it will need kind of very high sequencing depth to be sure about the consensus read, and can call the variant more confidently. And recently, for the human genome, HiFi has enabled the first complete genome of the human. This project is called the T2 telomere to telomere consortium, which can complete the remaining 8% of human genome that left unsolved by the initial human genome project in early 2000. Like at BMK Gene, we have done like many de novo genome assembly cases, which featured in a very high prestigious journals like Nature Genetic Plants, Molecular Plant, and Science as well. So this slide shows that the long read sequencing at BMK genes can help the de novo assembly in many cases. Next, let me introduce about the sequencing of the full-length RNA transcript. I believe that many of us know the RNA-seq technology already. That this based on the short read sequencing, which can sequence like any transcript from the samples. However, in eukaryotic species, our transcriptome can be more complicated because one gene can produce several isoforms due to the alternative splicings, and there will be a kind of complication during assembly of the sequence. Like the cartoon in the left hand side, like we start from the transcriptome. The scientists want to get everything to from the transcriptome data. So he started from the the book into small pieces, and then sequence each of them, and then he asked his team to help incorporate them into the original book, which there could be like the misalignment, misassembly can happen as well because of the isoform differentiation. So that's why the full-length transcriptome sequencing has emerged. So the long read by PacBio can have the better resolution in terms of alternative splicing, because the long reads can span in the full length of the transcript, so they can capture all the splice junction and no assembly required. And also they can detect like the alternative polyannulation as well. Polyannulation involved in the like the turnover rate of the messenger RNA. So like longer poly A tail can have the kind of longer turnaround lifetime of the transcript, but shorter poly A tail of the messenger RNA would have the shorter lifetime. Other application would involve like the identification of fusion transcript and isoform variant facing, and long non-coding RNA identification can be done more easily on the PacBio. And this is some the bioinformatic workflows that we can be done from the long read sequencing of the transcripts. So we can identify like the readout of the full-length cDNA molecule from end to end, and we can differentiate different isoforms at the sequence structure, and also quantify the expression level in each isoform as well. And the transcript will have the high accuracy and integrity, and this is compatible in many species, and can be done without the reference genome as well. And I think this is the last application I want to talk today, the metagenomic sequencing. In the microbiome research, many of us working with the 16S, 18S, ITS region of the microbial genome. These genes have the very long long sequence, like up to 1.5 kb or 1.8 kb, and they have like the variable region. The short read sequencing usually pick up only few region to do the sequencing, so they wouldn't get the full length of that the ribosomal DNA genes. Uh, in the recent study, they do the in silico experiment to check if they analyze only different fragment length of the ribosomal DNA, uh, how many or how much taxon can be identified in the microbiome. So the more rare is mean more unclassified species. Like if we do only V1 or V2 or V4 alone, there will be many taxonomy remain unclassified. But if we do the full length from V1 to V9, we will get a kind of very clear picture. So like almost zero become unclassified. So we can see that the taxonomic annotation using full-length ribosomal DNA sequencing will be able to identify the microbial species up to like most of them to the species level. Like right here, if you use like the old NGS technology, most of annotation will get around like at the genus level like 78%, and very few can be annotated to the species level. But for the PacBio, we can get annotation rate up to 95% for the genus and 60% at the species level. So that means if we do full-length amplicon sequencing on PacBio, we can identify the species of the in the microbial community of our sample more nicely. And this is the workflow of amplicon sequencing technology that we use. And sorry, this is the bioinformatic pipeline. From the raw data, we can provide data QC, feature sequences, taxonomy annotation at different level, like from phylum to species level, depending on the platform, and species diversity, like alpha, beta diversity. We can also do intergroup statistics. We can compare different combination between the treatment group. And also we can analyze environmental factor association with the samples. And here are some visualization graph that can we can offer by our bioinformatic pipeline. And most, I think most of them you are familiar with the publications associated with the microbiome study. Okay, and last but not least, high quality microbial genome. Like we have said that in the many genome projects, we often use many type of technology of different platform. If we want to get a very high quality or reference quality genome of your microbial species, we can do like use a different sequencing strategy like ONT read combined with NGS read, or we just do 30x PacBio CCS read with the NGS data to help polish. So you can see that this will provide like the zero gap of the bacterial genome. This also includes the plasmid as well. And for the fungal genome, we can get like the chromosome level genome assembly. We can combine like ONT, NGS, and PacBio, and also HiC data. So like the HiC data can help to grouping different contigs into the single chromosomes. Okay, in summary, PacBio HiFi sequencing method can yield very high accurate long read sequencing dataset, which can have a high long read like 10 to 25 kb with accuracy up to 99.5%. And a HiFi read can provide like comprehensive detection of all variants in the genome, both SNP, indel, structural variant, or inversion, and so on. The PacBio sequencing can have the application for the RNA sequencing. It can do the long range RNA sequencing, which offer superior isoform resolution, and full length amplicon sequencing as well, which allows the species to strain level identification. And I also would like to announce that at BMK Gene or Biomarker Lab, we just purchased a new sequencer from PacBio. It's named Revio, which is the latest PacBio sequencers at the moment, and it have like much higher throughput than the previous one. Can do like 1,300 human genome per year, which is 15 times higher than the previous model. This due to the very high demand for the PacBio sequencing, so that's why BMK Gene decided to purchase this sequencer. Okay, so lastly, we are having the year-end promotion up to 40% off for the sample arriving BMK Gene by December 20th. So if you have like the samples to sequence or you have the project in your mind, feel free to contact us via this email. I put my email here. Or you can also contact us through the official website as well, bmkgene.com. And thank you very much for your coming as well. Okay, Emma, over to you. Okay, so if there are any question, feel free to put your question in the Q&A box. Or you can raise your hand if you want. I'm happy to take question in Thai and English as well. Hey.
Thank you, Wilson, for the excellent talk. And do you have any questions on the webinar? Okay, if you don't have any questions about today's webinar, and we will send you a recording of today's webinar. And then if you have any questions on genomic or transcriptome or microbiome sequencing, and you could also come to our website or come to Wilson, and we will send the contact information to you, and it will be okay for you to contact with us in Thai or in English. Thank you for your attending. Thanks, Wilson.
Thank you very much.
Day. Bye.
Bye.